Dear students, அனைவருக்கும் வணக்கம், வந்தனம், நமஸ்காரம். Dr.N.Sathyamoorthy from Trichy. Today, I'd like to give a brief idea regarding plant tissue culture and the one more questions asked in your book. I will discuss one more questions as well as answers. Plant tissue culture is nothing but developing a new organism from a single cell in the lab condition. So developing a new organism from a single cell in the lab condition. So labல ஒரு செல்லுலேருந்து ஒரு ஆர்கானிசம் ஒரு ட்ரீயை நம்ம ப்ரொடியூஸ் பண்ணுவோம்னா ஆர் ஒரு பிளான்ட்டை ப்ரொடியூஸ் பண்ணுவோம்னா the concept is said to be plant tissue culture. Is it possible? Yes. Each and every plant cell has got the ability to produce the entire plant body. That ability is said to be totipotency. The basic concepts of tissue culture are totipotency, differentiation, dedifferentiation, and redifferentiation. Conversion of the meristematic tissue into a permanent tissue is said to be differentiation. The conversion of the permanent tissue into a meristematic tissue is said to be dedifferentiation. Once again, the conversion of meristem into a permanent tissue is said to be redifferentiation. Okay, into permanent tissue is said to be redifferentiation. And you know totipotency. So the basic concepts are totipotency, differentiation, dedifferentiation, and redifferentiation. And for tissue culture, you need the explant, the cells to be grown in tissue culture. So explant, for example, you are going to grow a particular cell. The cell is said to be explant. It may be a cell or a tissue or a bud or any part of the plant. For the culturing of this cell, we need a vessel. The vessel is said to be the culture vessel. It may be either a test tube or conical flask or petri dish or anything else. And for the growth of the cell, it needs nutrients. And that is a culture medium for the growth of this particular cell. Culture medium means the medium which furnishes all the nutrients needed for the development of a cell. For example, here the culture medium is present. MS medium, white medium, niche medium, Gampheric medium are the various medias used in plant tissue culture. And the most commonly used medium is Murosaki's Scooge medium. Okay. Now we go to the questions. I will discuss one by one more question. Okay. Question number 1 for you. Wait for a while. I'm learning technology. Question number 1 for you. Totipotency refers to. Just a minute before I explain this, the totipotency means the ability of the cell to produce the entire organism. The first option is capacity to generate genetically identical plants. The capacity of the cell to generate genetically identical plants. The genetically identical plants are called as the clones. The process is called micropropagation. So this is not correct answer for the question number 1. Capacity to generate a whole plant from any plant cell or explant. Yes, this is the correct answer. Each and every plant cell has got the ability to produce the entire organism. This is called the totipotency. Capacity to generate hybrid protoplast. This is incorrect. You know, protoplast, cell without cell wall. Plant cell without cell wall is said to be protoplast. Isolated protoplast of two similar species or two different species or two different genera fused to produce a hybrid cell. The process is called protoplasmic fusion or somatic hybridization. So this is incorrect for this question. Then recovery of healthy plants from diseased plants. Recovery of healthy plants from diseased plant means then after applying the chemicals or biopesticides, so we'll get a healthy plant. From the diseased plant, this is not correct. So the correct option is option B. Answer check panidoma. Yes, we will check it. Yes, totipotency refers to the capacity to generate a whole plant from any plant cell or explant. Okay. Next question for you. A second question for you. Micro propagation involves அப்படின்னு கேட்டிருக்கான். Micro propagation அப்படின்னா உனக்கு தெரியும். Tissue culture is also known as micro propagation. Plant tissue culture. Vegetative multiplication of plants by using microorganisms. We use microorganism for the veg propagation of plants. It is not a correct definition for this micropropagation. Vegetative multiplication of plants either by cutting or layering or budding, various kinds of veg propagation methods. We can use microorganism for the growth, not for this methodology. Vegetative multiplication of plants by using small explant. Yes. Explant, you know, the cell or the tissue or the bud to be grown, or the plant pot is to be grown in plant tissue cultures technique is said to be explant. So this is the correct answer. Vegetative multiplication of plants by using microspore. Microspore is the end product of meiosis. It's a gametic cell. So this is incorrect. Microspores, how the microspores are formed, you know. The microsporangium in the anther possesses microspore mother cells which are diploid in nature. The diploid microspore mother cell undergo meiotic cell division to produce haploid microspores in tetrads. From the microspores, male gametes are formed and it's involved in sexual reproduction. Then non-vegetative multiplication of plants by using microspores and megaspores. Microspore and megaspores are involved in sexual reproduction. So this is also incorrect. The correct option is option B. Check பண்ணிடுவோமா? Okay. 1 minute. So the correct answer is vegetative multiplication of plants by using small explant. Explant may be a cell or a tissue or a apical bud or axillary bud or the apical meristem or stem or a small portion of the flower or the anther for haploid plant production, like that. Okay. Next is match the following. Question number 3 for you. Match the following. Match பண்றப்பே நம்ம கரெக்டா பார்த்துட்டு ஆன்சர் என்ன வரும்னு சொல்லிட்டோம். First, totipotency. Totipotency means property of living cells develops into the entire organism. So இதுக்கு C option வரும். 1க்கு C option. De-differentiation. De-differentiation அப்படின்னு கொடுத்திருக்கா. Differentiation meristem into permanent தெரியும். Permanent tissue into meristem தான் அது வரும். அதாவது de-differentiation means differentiation. De-differentiation means reversion of mature cells into meristem. Meristem into permanent tissue, differentiation வரும். Permanent tissue into meristem, de-differentiation வரும். Explant, you know, selected plant tissue transferred to culture medium. So option D வரும். 3க்கு option D இங்க வருது. 2க்கு option A இங்க வருது. Differentiation means the biochemical and structural changes of cells leads to meristem into permanent. இதுக்கு B வருது. அப்ப இந்த கொஸ்டினை பொறுத்த வரையும் எது வரும்? CADB, that is A option வரும். நம்ம செக் பண்ணிடுவோம் எதுக்கும். These are all your book back questions. Answer is option A, C, totipotency, property of living cells develops into entire plant. De-differentiation, reversion of mature cells into meristem. Explant, selected plant tissue transferred to culture medium. Differentiation, the biochemical and structural changes of cells. Okay. Next question for you. The time duration for sterilization process using autoclave is dash minutes and the temperature is dash. You know what is sterilization? தமிழ்ல நுண்ணுயிர் நீக்கம்னு சொல்லுவோம். Sterilization means get rid of microbes. The removal of microorganism from the surface of the explant, culture vessel and culture medium. The explant is sterilized by using chemical sterilant like sodium hypochlorite, calcium hypochlorite and mercury chloride. Culture vessel and culture medium sterilized by using the instrument called the autoclave. In the autoclave, the vessel and the medium placed at 121 degrees Celsius for 15 to 30 minutes. So option B is the correct answer. The remaining 3 is incorrect. Anyhow, we will check it. Here the temperature is incorrect. Here the temperature is incorrect. Here the time duration is incorrect. Okay. The answer check panniduvoma. The correct answer is the time duration is option B, 15 to 30 minutes, and the time duration is 15 to 30 minutes. The temperature is 121 degrees Celsius. Next question for you. Which of the following statement is correct? இங்க இருக்கிற statementல எது correctனு கொடுத்திருக்கா? Okay, பாப்போமா? First statement is agar is not extracted from marine algae such as seaweeds. Marine algae, mostly, red algae, agar-agar, the gel-like non-nitrogenous substance extracted from the cell wall of rhodophycean members, Gracilaria gerardiana. Actually, you read this concept in the 11th standard plant kingdom. Economic importance of algae, we get agar-agar, gel-like non-nitrogenous substance extracted from the cell wall of seaweeds, rhodophycean members, red algae members, Gracilaria and gerardiana. Actually, agar is extracted, but here they mentioned not extracted. So this is the incorrect statement. It is extracted. And in plant tissue culture technique, this agar-agar used to solidify the culture medium. Now, culture medium means the medium which furnishes all the nutrients needed for the development of a cell. Culture medium may be a liquid medium or solid medium. If the liquid is converted into solid, we use this agar-agar. So it used as a solidifying agent in plant tissue culture technique. A callus undergoes differentiation and produce somatic embryoids. Yes, correct. You know callus. Callus is nothing but the undifferentiated and organized tissue formed during tissue culture technique. And from the callus, a bipolar embryo is formed with a radical, plumule, and cotyledons. The embryos developed from the somatic cells are called the somatic embryos or somoclonal embryos or embryoids. The callus is undifferentiated, but the embryo callus, callus is a undifferentiated and organized tissue. It is undifferentiated. But somatic embryo means the embryo with the radical, cotyledon, and the plumule. It is developed from the somatic cells. We are calling it a somatic embryo. It is due to differentiation, meristem into permanent. Then surface sterilization of explant is done by using mercuric bromide. Surface sterilization means the sterilization of the surface of the explant by using sodium hypochlorite, calcium hypochlorite, and mercury chloride. So this is incorrect. pH of the culture medium is 5 to 6. It is not 5 to 6. It is 5.6 to 6. So this is also incorrect. This should be 5.6. So option B is the correct answer. Anyhow, we check the key, the answer. Okay. Yes, the answer is option B is correct. Callus, the undifferentiated mass, undergoes a differentiation and produce somatic embryos, also called the somatic embryoids. The embryos developed from the body cells. The embryos developed from the somatic cells. We are calling it a somatic embryo. And then normal embryo is the end product of gametic fusion. We are calling it a zygotic embryo. Next question for you. Select the incorrect statement. See here, this is very important. Correct or incorrect? The question is incorrect statement from the given statement. A tonic used cardiac arrest is obtained from digitalis purpurea. Yes, it is correct. Digoxin. It is used to treat a heart attack, cardiac arrest, Medicine used to treat rheumatic pain is extracted from capsicum annivum. It is also right. The medicine is capsaicin. The anti-malarial drug is isolated from Cinchona officinalis. It is also correct. Malaria caused by the plasmodium and a female Anopheles mosquito act as a vector. In zoology, you read this concept in Human Health and Diseases. Malaria The causal agent is Plasmodium. It's a vector-borne disease. Female Anopheles mosquito carries the pathogen. Okay, it is a vector-borne disease means the pathogen carried by one organism to the other. Vectors. In biotechnology, we use the term vector, which carry the gene from the donor to the host cell. Okay, this is also correct. It is used to treat malaria. Then next is anti-carcinogenic property is not seen. Actually, it is seen in Catharanthus roseus. So this is incorrect. Catharanthus roseus, the vincorosea, yields vincristin and vinblastine, two alkaloids used to treat cancer. So anti-carcinogenic property seen in Catharanthus roseus, not seen is incorrect. Let me check. Answer check பண்ணுவோமா? Sorry. Answer is option B, anti-carcinogenic property is not seen in Catharanthus. Actually, it is seen in Catharanthus, so this is incorrect. Next question. Virus-free plants are developed from? Virus-free means they are free from viruses. Organ culture helps to produce a new plant. Protoplast culture used to produce somatic hybrids. Cell suspension culture, we are culturing the cells in a liquid medium. The correct answer is meristem culture, because the meristem, meristose means divisible. The cells are always in a state of cell division or continuous state of division. So viruses are enabled to grow in the meristematic region. So meristem or meristem culture helps to produce virus-free plants, or the plants free from viral infection. This is the correct answer. நம்ம செக் பண்ணிடுவோம் எதுக்கும். Okay. Yes, meristem culture is a correct answer. You know, 3 kinds of meristem reported in plants. One is the apical meristem. You read in Levin standard, tissue and tissue system. You read the meristem. I will explain. It is very useful for your competitive examination. One is meristose means divisible. The cells are always in a state of cell division or continuous state of division. 3 kinds of meristem reported in plants. One is the apical meristem. It is seen in the apices of root and stem, or root and shoot tip possesses apical meristem. Intercalary meristem actually seen in the internode. The distance between 2 successive nodes is said to be internode. Here it is present, and it is also seen in the leaf bases of monocot plants. And in monocot, it is responsible for leaf sheath formation. Lateral meristem seen on the lateral sides of the root and stem, and it is responsible for secondary growth. So apical meristem, location root and shoot apices, intercalary meristem seen in the internode and leaf bases of monocot. And lateral meristem seen on the lateral sides of the root and stem. Apical meristem responsible for elongation of root and stem in its longitudinal axis. Intercalary meristem responsible for internode elongation and leaf sheath formation in monocot. You know, monocot plant leaf like banana. The leaf base is modified into leaf sheath. So sheathing leaf base, characteristic feature of monocotyledonous plants. The leaf sheath, this is leaf sheath. And the lateral meristem responsible for increase in thickness, increase in girth of plant. Okay, you read these concepts in Levin standard, tissue and tissue system. Okay, next question for you. The prevention of large-scale loss of biological integrity. 4 options are given. One is the biopatent. Second is the bioethics. Third is the biosafety. And fourth is the biofuel. Before going to check the answer, first we should clearly know the concepts. What is biopatent? What is bioethics? What is biosafety? And what is biofuel? I'll explain one by one. Okay. First, we go to biopatent. Actual definition is biopatent means patents of living organism. It may be a plant, it may be a microbe, or it may be animal. Plant or microbe or the animal, and related biological and biotechnology enabled invention. So using biotechnology or biology, you invent something from the plant or microbe or the animal, or you are modifying its genetics by using genetic engineering or biotechnology, you need a patent. If you get patent is nothing but a special right to the discoverer. For example, you produce a transgenic plant, you produce a transgenic animal, genetically modified organism, then you have a special right to that invention that has been granted by the government through legislation for trading new articles. For trading and for new articles. So if you invent a new one through biotechnology or any process, then you have a right to sell that particular product or particular organism. This is called the biopatent. Okay. Next is the bioethics. A simpler definition is the ethical issues emerging from advance in biology or medicine. Advancement in biology, advancement in medicine leads to many modifications in the organism. We are genetically engineering plants, we are genetically engineering the animals, we are genetically engineering the microbes. So it may lead to some ethical issues. So the ethical issues emerging from advance in biology and medicine is said to be bioethics. Or bioethics, the study of ethical, social, 1 minute, ethical, social, and legal issues that arise in research. So you are doing a research, you are producing a new organism, modifying an organism. It may lead to some ethical issues or social efforts, some legal issues. It is said to be bioethics. Take very simply defined, medical, animal, and plant-related research causes some ethical, social, and legal issues. It is said to be bioethics. We should maintain ethics on this. Then next one is biosafety. We are using a lot of microbes in the hospital, in the lab. It may be harmful to the environment. So we need safety. That safety is said to be biosafety. Biosafety means safety of the lab, the research lab where they are doing research in microbes or other organisms. Hospital workers, because hospital is enriched with the pathogens. Various patients are coming to the hospital with infection. For example, some may have tuberculosis caused by Mycobacterium tuberculosis. Some have pneumonia, Diplococcus pneumoniae. These are airborne diseases. Some diseases are airborne, some diseases are waterborne. So hospital environment is always a pathogenic in nature when working with potentially hazardous, toxic, and pathogenic microbes. In that case, we need safety. That safety is said to be biosafety. The prevention of large scale, it is not large, this is large scale, LARGE, large scale loss of biological integrity, focusing both of them. So we are working in a lab, hospital, or in an environment where toxic pathogenic microbes are seen grown, we need safety. That safety is said to be biosafety. Okay. The next one is the biofuel. The fuels obtained from living organism or plants or animals, especially here from algae. The fuel produced from biomass, which biomass? Algal biomass. The organism used in the production of biofuel is Botrycoccus bruni, used to produce algal biofuel. So these things are asking your question. The correct option is actually, I think, biosafety. Let me check. Oh, sorry. So biosafety is very important. Large scale, see, wait for a while. Learning, still learning. Okay. Large scale loss of biological integrity அப்படிங்கிறது we need biosafety is the correct answer. Okay. Next question for you. Wait for a while. Cryopreservation means it's a process to preserve plant cells, tissues of organs. Tissues vechikalam and also organs vechikalam. So cryopreservation, it's a process to preserve plants, plant cells, tissues and organs, anything else, or any plant parts. At very, yes, this is correct. Cryopreservation, preservation, it's a kind of preservation. It is correct. Options enna koduthirukka. At very low temperature by using ether. We are not using ether in cryopreservation. At high temperature by using liquid nitrogen. We use liquid nitrogen only in low temperature. At very low temperature of minus 196 degrees Celsius by using liquid nitrogen. This is the correct answer. At very low temperature by using liquid nitrogen, here the temperature is not mentioned. So this is the correct answer. So cryopreservation means preserving the germplasm in liquid nitrogen at minus 196 degrees Celsius. This is the correct answer. Let me check. It helps in germplasm conservation. Yes, cryopreservation at very low temperature of minus 196 degrees Celsius. Here I should mention 196 degree Celsius. Degree Celsius by using liquid nitrogen. This is correct. Okay. Then next question for you. I hope it may be the last question. Solidifying agent used in plant tissue culture. So already the same question asked in a different way. Solidifying agent used in plant tissue culture is agar-agar. Agar-agar, the gel-like non-nitrogenous substance extracted from the cell wall of red algae, rhodophysian members used as solidifying agent. What is the role of these other things? Nicotinic acid, cobalt chloride, and EDTA. We see one by one. Before that, we will check the answer. Yes, agar-agar. Solidifying agent used in tissue culture is agar-agar. Then what this EDTA represent? EDTA, E for ethylene, D for diamine, T for tetra, A for acetic acid. It helps to prevent DNA and RNA degradation during genetic engineering. Then cobalt chloride or cobaltous chloride, it is an allergen for some people. You know, allergy, inappropriate excessive response to the external stimuli is said to be allergy. The chemicals causing or the substances causing allergy are called allergens. So it may be an allergen. The nicotinic acid protect the plant from stress. So stress relieving hormone or prevent the plant cells or plant from stress. So these things you know. Thank you very much for hearing, students. Thanks a lot. The next class we will see 2 and 3 more questions in this concept. Thank you. Thank you. Thank you. Tata bye bye. See you.